my name is Chandra Etheridge, and today I'm going to be showing you guys how to extract DNA from a banana cell. Can anyone in the audience please raise your hands and tell me what does DNA stand for?
Deoxyribonucleic acid.
Yes, DNA stands for deoxyribonucleic acid. DNA is inside of every living organism, including bananas. In order to extract the DNA from a banana cell, you'll need a Ziploc bag, a piece of the banana, about half of it, some type of filtration system, multiple cups, half of a cup of hot water with a teaspoon of salt, in which I've already mixed the salt in the water, and some cold isopropanol alcohol. This concentration is about 91% alcohol. The first thing you want to do is seal the bag. Make sure there's no air bubbles. And mush the banana around until it's the consistency of banana pudding. Like such. Next, you'll want to add the hot salt water mixture. And do the same thing. Make sure the bag is sealed so you don't get banana guts everywhere. What we're doing when we're mushing this down is we're breaking apart the cell membrane. The cell membrane is what protects each individual cell from the outside environment. It also allows things to enter and exit the cell. Next, you want to add about a teaspoon of any type of dish detergent. Not too much, because if you add too much, it'll create suds, ruining the experiment. And mush it around again. DNA is housed in the nucleus of each cell, but that differs for bacteria and archaeans. The DNA in bacteria and archaeans are housed in the cytoplasm. It should look something like this. Next, you want to get your filtration system ready. Be careful not to poke a hole in the bottom of the napkin, or it will allow big chunks of the banana to get through. We're gonna need a little bit more. Since DNA, it can only be seen microscopically, we're gonna have to figure out a way for us to see the DNA. And in order to do that, we're gonna take the cold isopropanol alcohol, and instead of directly putting it inside of the banana DNA mixture, we're gonna let it drip along the sides, creating a barrier between the water and the alcohol. You can add however many drops you feel necessary until you can see the separation of the water and the alcohol mixture. It's gonna take some time to form. Earlier I did this experiment and this was the result. Right here, you can see the DNA floating around. Since the human eye can only resolve up to about 20 micrometers, the alcohol allows the DNA to come to the surface so you can see the double strands and the helices. Right here is the DNA. As you can see, easy experiments can be done like this in any home kitchen to get the DNA out of any living organism.